Hi everyone, my name is Narmuratova. I'm Master of Science in Nutrition and Metabolism. Today we will talk about the biomolecules. First of all, let's start our today's discussion with the questions. What are biological molecules? The next question, what are four biological molecules? As we know, all the living organisms consist of four biological molecules: lipids, nucleic acids, carbohydrates, and proteins. Learning outcomes for today's class. As a result of this lesson, you will be able to define carbohydrates in terms of structure and function. Give examples of monosaccharides, disaccharides, and polysaccharides. Define fats and oils in terms of structure and functions. Provide explanation of their differences. Give examples of saturated, unsaturated, and polyunsaturated fatty acids. Define amino acids in their function. Name of amino acids. Essential amino acids, and provide an explanation of the reasons why called essential. And define nucleic acids in terms of its structure. Describe DNA structure in terms of number of base pairs, lengths, turns of double helix. Then compare DNA and the RNA molecules in terms of structure. Use the practice Chargaff's rule and explain complementary base pairing, and give an example of hydrogen bonds. Let's start with the carbohydrates. Carbohydrates or carbs are sugar molecules. The main building blocks of carbohydrates are monosaccharides. Monosaccharides consist of three atoms: carbon, hydrogen, and oxygen. The number of carbohydrates, the number of carbohydrates can be hexoses or pentoses. As you can see in here, hexoses consist of 6 carbon molecules. Pentose include 5 carbon molecules. One of the examples of hexoses are glucose. One of the main examples of pentoses are fructose. Carbohydrates classified as monosaccharides or simple sugars, oligosaccharides that consist of 2 to 10 sugar units, and the polysaccharides, more than 10 sugar units. Monosaccharides or simple sugars divided into groups depends on functional groups and the number of carbon atoms. Examples of the functional groups: aldoses, glucoses, ketose, and fructose. Number of carbon atoms: trivoses, butyraldehyde. Examples of tetroses, erythrose, pentoses, ribose, and hexose is glucose. Carbon atoms in here is 3. Here is 4. If the carbon atoms is 5, that's called pentoses. Carbon atoms is 6, in this case, hexoses. Oligosaccharides can be disaccharides, trisaccharides, tetrasaccharides, and pentasaccharides. If the two carbohydrates binds each other, they can be disaccharides. The main examples of them: maltose, lactose, and sucrose. If the trisaccharides, main examples of raffinose, tetrasaccharides, stachyose, and pentasaccharides. Biomolecules. If we talk about the polysaccharides, also divided in two groups, homopolysaccharides and the heteropolysaccharides. Homopolysaccharides, the main examples of starch, glycogen, cellulose, histidine, and inulin. Heteropolysaccharides, peptidoglycan, hyaluronic acid, chondroitin sulfate, keratin sulfate, and agar-agar. Next group of saccharides are oligosaccharides. As we said before, these saccharides formed from two monosaccharides are the simplest polysaccharides. Here shown two molecules of monosaccharides that binds each other and gives the form of disaccharides. For example, sucrose consists of two monosaccharides, glucose and fructose. If we talk about the lactose or milk sugar, it consists of galactose and glucose. Sucrose is a principal product of plant photosynthesis and is refined into common table sugar. The next class is polysaccharides. Polysaccharides containing dozens to hundreds of monosaccharide units can function as reservoir for glucose, as structure components, or adhesives that help hold cells together in tissue. One of the main examples of polysaccharides are starch. It's a primary storage carbohydrate in plant cells. If we talk about the animal cells, it can be in the form of glycogen, the most common storage carbohydrate in animal cells, actually in liver. And cellulose, the major constituent of plants cell wall. For example, here shown glycogen molecule. As we can see in here, all the main building blocks of glycogen are glucose units. The next question, what are the main function of carbohydrates? They are a source of energy, structural components of tissue, and regulates fat metabolism. The next main biomolecules are lipids. In this case, what are lipids? They are organic molecules with long hydrocarbon chains that are insoluble in water but soluble in organic solvents. Lipids also contain carbon, hydrogen, oxygen molecules. Lipids consist of fats, oils, hormones, and certain components of membranes that are grouped together because of their hydrophobic interactions. If we talk about the lipids classification, they are classified as simple lipids, compound lipids, and derived lipids. Simple lipids, esters of fatty acids with various alcohols that include fats and waxes. Compound lipids, ester of fatty acids with different groups of alcohols and fatty acids. They can be phospholipids, glycolipids, sulfolipids, aminolipids, and the lipoproteins. Derived lipids from both simple and compound lipids, but by hydrolysis. They can be fatty acids and cholesterol, Fatty acids also divided in two groups, saturated fatty acids and unsaturated fatty acids. Compound lipids, for example, phospholipids, they are alcohol and fatty acids, phosphoric acid plus nitrogen containing base. They keep the form of phospholipids. Glycolipids when the fatty acids binds with amino alcohol and carbohydrates. Aminolipids, they binds with fatty acids, alcohol, and amino containing groups. And the lipoproteins, when the proteins binds with lipids. What are fatty acids? Fatty acids are a long-chain carboxylic acid. The structure of fatty acids varies in one or more of three ways. The first one, the number of carbon atoms in the carbon chain. The next, the number of carbon-carbon double bonds they contain, or the position of double bonds. As the length of fatty acids increases, the melting point of the fatty acids increases. Here are given some examples of structure of fatty acids. As we said before, fatty acids can be saturated or unsaturated. Types of amino acids saturated, here, no double bonds, as shown in here. Monounsaturated, they have one double bonds, and polyunsaturated, multiple double bonds. The main examples of saturated fatty acids can be beef or animal fat, butter, and coconut oil. Here are no double bonds, and the structure is straight. They are solid at room temperature. Another form of fatty acids can be trans fatty acids. They have one or more double bonds in trans configuration and the straight structure. They can be solid or solid at room temperature. The main examples of them are margarine, cream, soap with puff pastry, and the chicken pie. So next one is monounsaturated, one double bond in cis configuration, band structure, liquid at room temperature. The main examples of them, olive oil, canola oil, and peanut oil. In addition, polyunsaturated, multiple double bonds in cis configuration, even more band in structure, liquid at room temperature. Main examples of them, soybean oil, corn oil, and fatty fish. Functions of lipids, they are biological. The main biological functions of the lipids are diverse as their chemistry. In the body, fats serve as the efficient source of energy and also stored in the adipose tissues. Phospholipids and the sterols are major structural elements of biological membranes, acts as functional component of the cell and provide the hydrophobic barrier and also activators of enzymes. What are proteins? Proteins are large complex molecules composed of amino acids. Proteins functionally depends on the molecular size, shape, and chemical properties. The main building blocks of proteins are amino acids that consist of carbon, hydrogen, oxygen, and nitrogen. Amino acids binds each other by peptide bond and gives the form of peptides. And peptides binds each other, they gives of special size of proteins. Amino acids is hydrocarbon that have central carbon attached to the amino group and hydroxyl group, and with the side chain or R group that varies among different amino acids. In nature, we know that proteins consist of 20 amino acids. Amino acids classified as hydrophobic amino acids, hydrophilic amino acids, and the special amino acids. Classification of amino acids are nutritional and based on R group. By the nutritional, they divided by two groups, essential amino acids and non-essential. Non-essential amino acids synthesized by our cells are essential amino acids derived from the foods. Based on R groups, amino acids can be non-polar aliphatic R group, polar uncharged R group, aromatic R group, positively charged R group, and negatively charged R group. Here are given essential amino acids: phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, leucine, lysine, histidine. All these amino acids not synthesized in our cells. Protein structure can be primary when the amino acid sequence in the form of polypeptide. Secondary structure is generated when hydrogen bonds between amino acids is close proximity to each other, form to regular structure alpha helix and beta plated sheets. Tertiary structure is a 3D shape of the polypeptide chain when it's folded. Quaternary structure is the interaction among multiple polypeptide chains. The main functions of proteins include support, for example, cellular cytoskeleton, metabolism, for example, enzymes and the hormones, protection, for example, our antibodies, communication, for example, signal transduction proteins that acts as messengers between and within cells, and the regulation, the transcription factors regulate transcription process. Here given some types and functions of proteins. For example, types of digestive enzymes: amylase, lipase, pepsin, trypsin help digestion of foods by catabolizing nutrients into monomeric units. One of the examples of transport proteins, hemoglobin or albumin, carry substances in the blood or lymph throughout the body. Structural types of proteins actin, tubulin, keratin consist different structures like a cytoskeleton. Storage type, legume storage proteins, egg white albumin provide nourishment in early development of the embryo and the seedling. What are nucleic acids? Nucleic acids are molecules composed of monomers known as nucleotides. Two types of nucleic acids, DNA, deoxyribonucleic acids, is large molecule, and RNA, ribonucleic acids, relatively small molecules. The main building blocks of nucleic acids are nucleotides, which consist of carbon, hydrogen, oxygen, and nitrogen, and also consist of phosphate, sugar, and a nitrogen base. Here, given nucleotide structure, which consists of nitrogen base, sugar, and a phosphate group. What is the difference between DNA and RNA? As we know that DNA or deoxyribonucleic acids consist of two strands and a double helix. RNA molecules, ribonucleic acid, single strand, and they are differ by nitrogen base. In DNA is thymine, cytosine, adenine, guanine, but in RNA, instead of thymine, they have uracil. And cytosine. Okay. It's all the topics that we talked, all the information that we talked today. As we said before, by the end of the session, students were able to define carbohydrates in terms of structure and function. All things we talked today. I hope you have more ideas and you have more examples for monosaccharides, disaccharides, and the polysaccharides. You can give an examples for unsaturated, saturated, and the polyunsaturated fatty acids. And also you're able to define amino acids and their function, and you can define nucleic acids in terms of its structure, and you can give the examples of Chargaff's rule and explain them. Thank you all of you for coming today's my class and joining for this lesson. If you have any questions, feel free asking or just texting me. So thank you. Bye.